Welcome back. We missed 2 weeks. One was due to my unexpected absence. Once again, sorry for that. And the other was due to the holidays. So welcome back after 2 weeks. This is a slide where we stopped last time. We'll continue from here and hopefully finish this chapter today and start the new chapter on gap junctions and calcium signaling by the second half of the course. Lecture, sorry. So, remember we were talking about the movement of the molecular motors over the microtubular network. Now we'll slightly talk about DNA and the relationship between the DNA and the microtubular motors and the channel capacity related issues. So, hybridization is the process of combining the complementary single-stranded nucleic acids to form the DNA. So we have the nucleotides on these nucleic acid strands, and these will typically bind with their complements on the other side under normal conditions. So if you have two perfectly complementary strands, these will attach together very nicely and form that famous double-strand DNA form. And this process is given the name of annealing. However, if there is a single inconsistency between these two strands, where you have the inconsistency between two nucleic acids that were meant to be complementary, then this binding between these strands will not be so much favorable in terms of the energetic relation in between. So annealing will not be that strong in that case if there's some mismatch. Now annealing may also be reversed if you heat up this two strands, double-stranded molecule of DNA, so that you can, what you do is actually you break the hydrogen bonds that form this hybridization, and this way these two strands will be separated from each other. And this is the inverse of annealing, and it's called the denaturation. A communication network now can be proposed based on the molecular motors, and this is given in the reference 21. At the end of the chapter you can find all those references. The key concept there is that the DNA hybridization now exerts a stronger binding force with longer complementary pairs. So as the length of the pairs increase, the force between the attractive force between these strands will be stronger. Now you try to benefit from this as follows. Now you have the motor transporter with a short DNA complementary strand and with enough binding energy to pick up the longer complementary strand of the information cargo. Okay? So the transporter itself has a shorter strand. And this molecular motor is carrying its cargo along the microtubule, and when it reaches its destination, there you will find a longer complementary strand. Since it's longer, it will have a more strong attraction with the cargo compared to the molecular motor, which is a shorter DNA complementary. This way, actually, the motor will arrive at the destination and unload its cargo at the destination. It will get rid of its cargo. So now the molecular motor is free to leave the microtubule rail. So it leaves the microtubule rail and floats with Brownian motion inside the cytoplasm, hopefully to reach at a place where it can get a new cargo and again get on the microtubule network to carry the new cargo. So that's actually what's happening, and all this is explained now in reference 22. The one important thing was large numbers of molecular motors traversing a complex microtubule network or an actin network where you have those actin filaments you pull or push, have the potential to carry a significant amount of information. The more motors you have, the more information you can carry. So still, in terms of biological cell signaling mechanisms, we don't know how many mechanisms are used actually in the living organisms and how these are actually working. We have some information on that, but still there's research going on that. But our focus in this chapter is on the intersection of molecular motor information transport and the communication network. How we can benefit from the molecular motors to carry information, and how much information we can carry with this. So these molecular motors can be engineered to work in vitro, which means inside the living cells, living organisms, in which case you can imagine the standard communication protocols will be utilized. Well, this is a little bit arguable, as we will see in the following slides. One of the main characteristics affecting the protocol will be the bandwidth delay product, because the bandwidth delay product defines how much information we can carry in the network at any given time. So the discussion we will have in these few slides will be something you're supposed to actually know from your computer networks courses, but let's try to refresh our knowledge on information transportation there. So the bandwidth delay product is just actually a multiplication of the bandwidth and the delay. And the important thing is this product defines the carrying capacity of the communication channel. In other words, how much information you can put on the channel at any given time. And it refers to the bandwidth of the channel. Okay, remember that's given in BPS. So how many bits you can carry in 1 second. So you can think, imagine the communication channel to be something like a water pipe. So the bandwidth will show you the width of that channel. Whereas the propagation delay is the time it takes to go from this end of that pipe to the other end. So if you multiply the bandwidth here, the width of the channel, with the length of the channel, then you will get the volume. Okay? And that would be the amount of water you will have in that pipe at any given time. If you look at it, if you have the analogy to the communication network, then that will be the number of bits you can have in flight on that channel at any given time. If the channel is wider, which means more bandwidth, then that means you can have more information in flight at any time. However, without making the channel wider, if you make the link longer, again you can put more information on the channel. But now the problem is, since the length is longer, that means the time it takes to go from this end to the other end of the link will be longer, which is not something we want so much, but it may happen. A very good example of that would be what? Something we often discuss, the bandwidth delay products, is the satellite channel, for example, where you have a very wide band and also a very large delay there. Okay? That's why the bandwidth delay product in the case of a satellite link, for example, would be very high. Okay? So the bandwidth delay product is now bits per second times second. So the seconds will cancel out, so it will be in bits, in units of bits. And if one thinks of the channel as a pipe, as we discussed, conducting fluid, this will be measuring the amount of fluid in the pipe when the pipe is completely filled.
So why are we concerned with this amount of?
Good question. Now, if you have a wider pipe, and that means I can send more water from point A to point B. So that means I have more capacity for transportation, right? However, when you consider this in terms of communications, remember that our channels are not error-free in real life. So a packet in flight, that means a packet that is being at the moment transmitted from point A to point B, but it has not been completely received at point B, might end up to be transmitted to B successfully or not. If not, I have to find a way of sending it successfully. A typical method will be, as we will soon discuss, repeating the packet, retransmitting in other words. Okay? So in order to be able to retransmit, I have to store that information. It's not like send it and forget it. You cannot forget that packet until you're sure that it has been successfully transmitted. Now, if the channel's bandwidth delay product is large, that means the number of packets I need to remember is very large. Okay? So that means I need a lot of buffering on the sender side. If I want to fully utilize the channel. Now it is possible that I have a very wide pipe, but I'm just sending a very small amount of water. Okay? That's also possible. You don't have to fill the pipe. But in that case, that means your utilization is very low. We would like to utilize the channel as much as we can. But if you do that, as I said, then when you think about data communications, you should have a method of, a way of storing all that information at the sender side, because you may need to retransmit. That means huge amounts of buffering. Okay? So now, what happens when we do this, for example, in molecular communications? How do you store all that information at the sender cell? You have to think about that. Also, you should be able to, for example, differentiate between the packets. Okay, you realize that a packet was damaged. So you refer back to your buffer. How do you find the correct packet in that buffer? Okay, so these are problems that need to be addressed. In human engineered networks, to cope with these errors, we apply the automatic repeat request, which is in short known as ARQ. It's a family of protocols. There are many variations of ARQ, okay? But with similar ideas in mind. ARQ is typically one of the simplest and most effective protocols for coping with errors. It provides both error detection and retransmission to make sure that the information is sent to the destination correctly. The simplest version of ARQ would be simple stop and wait protocol. Okay? This corresponds to a window of width of one packet. Okay? You send one packet and you stop and wait. That's where the name stop and wait comes from. Wait for what? Wait for an acknowledgement from the receiver side saying that, yes, I have successfully received it. Now, the reason you may not receive this acknowledgement could be various. One, the packet has been lost. So in that case, the receiver was not even expecting a packet. So the receiver cannot say, I didn't successfully receive it. He or she is not even aware of that. Okay? It might be sent, but it might be damaged. So the receiver receives it, but realizes that there's something wrong with the packet. In that case, the receiver knows that there has been a packet, but it is damaged.
In this case, it also sends acknowledgement?
Depends on how you implement it. You can send what we call a NAC, negative acknowledgement, saying, I failed to receive this. Or you might simply just ignore it, because typically the transmitter starts what we call a timer. So you expect the acknowledgement to come in some given time duration. If you do not receive the acknowledgement in time, then you retransmit. In that case, you don't need a NAC. But having a NAC makes the protocol faster. You don't need to wait till the end of the timer, you may immediately retransmit. But there are cases where you realize that there's something wrong, but you cannot send an ACK. Why?
Because maybe the header information, you may not know from whom the packet is coming from, so you can.
Right.
You don't know the transmitter.
If there's an error, but the error hits the header, in that case, you know there's something wrong, but you either don't know, as your friend said, from whom the packet came, so you cannot inform the sender, because you don't know the sender. Or you know the sender, but for example, to be able to differentiate between the packets, you will have the sequence numbers. The sequence number is damaged, so you know who sent it, you know it is damaged, but you're not able to say, this packet was damaged. Okay? So, there might be various reasons for failures. Or, yet another one is, acknowledgement packet is itself lost. So, the packet was transmitted successfully, but when you send the acknowledgement, that acknowledgement is either lost or damaged. In that case, again, you cannot identify, and you have to retransmit. But note that these retransmissions will cause, in that case, may cause duplicate packets in case of failures in acknowledgement. Okay? The original was received correctly, but acknowledgement failed, so I retransmitted. So the receiver now has 2 copies of this, and you have to detect that they're duplicates and ignore. Take only one and ignore all other duplicates. You might have multiple duplicates. Okay? So these are the basic ideas, as you will remember from computer networks courses. So if we're using the stop and wait protocol, it's a reliable protocol, because if something is lost, you retransmit. But it's not very effective. Actually, it is effective, but it's not very efficient. I should correct it. So a message is sent, and the sender waits for an acknowledgement before sending the next message, the next packet. If no acknowledgement is received in that timeout duration, then we have a retransmission of the lost or damaged packet. If the acknowledgement is received, then the next message can be sent, and the acknowledgement now acts as what we call a simple feedback mechanism to the sender. Okay? Now coming back to the molecular motor systems. What if I apply these ideas? By the way, before going into that, there's one important thing. This word reliable here. This is reliable. This protocol is reliable because if some packet is lost or damaged, you insist on sending it successfully. One important thing is, do we always need this? Do we always need reliability? Let me take the question this way. When I use the term reliability, in terms of computer networking, data networking, what does this remind you? The term reliability. It should ring the bells. What do you remember when I say a reliable protocol?
Awake all the time.
Tell me a reliable protocol.
TCP.
TCP. TCP is known for insisting on reliability. Do we always need TCP then?
No, if you have some real-time communication, maybe you might drop reliability in favor of minimized delays or something.
Very good. So the TCP protocol is there to provide reliability, but there are cases where we don't require so much reliability. So as you said, real-time applications, like what?
Like video chat.
Video, okay. Video transmissions, for example. Note that this is actually a soft real-time, we should say. It's not a hard real-time, but it's a soft real-time transportation application. Now, when was TCP proposed? How old is TCP? Do you have any idea? No one remembers? Was it before you were born or before I was born?
Between us.
Between us? Bless you. Or was it before Facebook? When was Facebook invented? Was it before you were born or after? After, okay. TCP was out there when I was something like 3-4 years old. So you're right, between us, but very close to my birth. It was in the first half of the 1970s. So when TCP was out there, no one thought about video applications, for example. The purpose was to be able to connect networks together so that you have the internet, network of networks. And there, the applications we could think of was simply messaging, but I don't mean video messaging, I mean text messaging, emails, and file transfers. There you need reliability. So these networks were designed with reliability in mind. However, things have changed in time. Now we have the video applications, and TCP is not a good fit for that. Even UDP itself, such a simple protocol without any reliability, is even better for video applications. Or you have RTP, RTCP together to provide communications. Now, if you're taking the concepts of 1970s and trying to apply them directly to nanonetworking, you might fail. You may need to consider, pay attention, I say may. You may need to consider applications where reliability is not so important, but other things might be more important, like transmission in time, or higher bandwidths, or whatever, okay? Don't forget that we're not trying to do file transfer between the cells in the human body, okay? And you might be able to tolerate some error. Like, assume that I'm trying to transmit the body temperature or the glucose level. If a minor mistake in the transmission causes the body temperature to be 0.01 centigrades higher or lower, that's not a big deal. Okay? We wouldn't be too much concerned about that. However, if a single bit error might cause the body temperature of 37 degrees to be transmitted as 58 degrees, then that's a major problem. Okay? So, see, it depends on which bit has been erred. So if I can find a better modulation scheme, or encoding, let me say, in better terms, in which a single bit, or maybe a few consecutive bits, cannot change the value so much. If I can somehow bound the error, then I might drop reliability, as long as my error is confined in some given range. Okay? So try to think outside the box. Do not always take the ideas we already know from human scale networks and try to translate them to the nanonetworking world. Maybe you don't need them, or maybe they're not so effective. Okay? So, considering molecular motors, motor systems, now we have our messages or packets in the form of chemical substances, molecules. And these will be now attached onto the molecular motors. Typically, you don't attach the molecule itself directly on the molecular motor, but you put it in a vesicle and attach the vesicle on the molecular motor. So, as we have already discussed, there is a probability that the molecular motor may be detached from the rail, which means it will not be able to reach the destination, or at the crossing points, it may take the wrong path and go to the wrong destination. So our messages may not arrive at the destination at all, or they may be too much delayed. Okay? So So we have to take care of this. So a straightforward application of what we already know could be, for example, the stop and wait protocol into this mechanism. But we will see that it can be very inefficient. We know that if the bandwidth delay product of the channel is high, then much of the channel is wasted if we're using the stop and wait protocol, because this is equivalent to having that very wide channel, but you're sending a very narrow stream of water in that pipe. Okay? You're not fully utilizing the pipe. Why? Because you have to, as the name implies, you have to stop and wait. Send one packet. You cannot send the next packet until that packet has gone all the way through the pipe. It has been processed there, acknowledged, and the acknowledgement comes all the way back to the transmitter. You cannot put the next packet in the pipe until this happens. Okay? So if you consider now the molecular motor system with the stop and wait protocol implemented on it, the bandwidth in terms of packets per second, if you consider that, where the packet is one motor cargo, you put the cargo on the molecular motor. Now that molecular motor has to walk all the way through the microtubule, unload its packet, either that one has to wait or another molecular motor typically, because the same molecular motor cannot go back and forth. That one detaches. Meanwhile, the acknowledgement is prepared. Another molecular motor that will walk in the reverse direction is found, and the acknowledgement is placed on the microtubule and comes back to the source. All this has to be done for stop and wait protocol to work. Again, as we discussed before, if the acknowledgement motor is lost because it is detached or takes a wrong path, then that again appears to be a lost packet, although the packet has successfully arrived. We should make use of sequence numbers to differentiate between the messages so that you know which one was damaged. So this will allow us to take care of the lost and delayed packets and their acknowledgements. So what about the efficiency of using stop and wait using the microtubular network? The efficiency can be defined in terms of the round-trip transmission. Round-trip is a time starting from the moment you send the packet from the sender, it goes to the destination, processed there, acknowledgement is prepared and sent, and the acknowledgement arrives at the source again. So the two-way transmission is altogether called the round-trip transmission. So if we have the following notation as n sub f for the number of bits in the information frame, n sub a similar for the acknowledgement, r is the bit rate of our channel, t propagation is the propagation time of the message, either the information or the acknowledgement, from the source to the destination one way. Processing is the processing time for the packet. Let's assume same processing time for the acknowledgement and the information package. And TF is the time to receive a frame. It arrives at the destination, but it takes time to take it in. Then this round trip time would be twice the propagation time, because first the message goes and then the acknowledgement comes all throughout this link. Twice the processing, one for the frame, information frame, one for the acknowledgement. Time to receive the frame and time to receive the acknowledgement. Okay? Some of these. And the time to receive the frame can be also rewritten as the number of bits in the frame divided by the R, the rate, because the longer the packet is, it will take more time to put it on the link. So it is dependent on this number. Similar for the acknowledgement. Okay? Now the effective transmission rate then can be defined as in here. The number of bits we're delivering, which will be the number of bits in the frame minus the amount of overhead. Because the frame I'm sending does not contain only the data, but there's also something more in it. Typically what we mentioned as a header. The source identifier, the destination identifier, maybe a sequence number, maybe some more additional bits for redundancy against errors, like the CRC checks or others. So you have to subtract that overhead to find, here the numerator is the number of useful data bits divided over the time it takes for the round trip transmission. Because only after T0 you can put the next packet on the link. Okay? So that would be our effective transmission rate. Then we can define the efficiency of the protocol as the effective transmission rate over the raw data rate of the channel. In the raw data rate, we are not differentiating between useful bits and the overhead bits. We are not differentiating between the time you're sending really a frame or a bit, or you're avoiding from transmission until, for example, the acknowledgement arrives. You were able to send a bit, but you didn't do that because you're still waiting for the acknowledgement. In the denominator, we don't take that into account. That's your rate, the raw rate. Whereas in the effective one, you take that into account. And this ratio gives you your efficiency. Substituting what we had in equation 22 in here for T0, and with some simple arithmetics, you can end up with this ratio. Everything here is what we already have discussed. Okay? And here, twice the propagation time times the rate gives us the bandwidth delay product we discussed. Okay? So that would be round trip time times your data rate, raw data rate. You can have better versions of ARQ, like go back N and selective ARQ. Do you remember what these were? In the case of go back N, you sent out one packet without waiting for that to be acknowledgement, as in the case of stop and wait. You send the other one. So now you have 2 packets in flight. And then the third one, fourth one, fifth one. You can send many packets, but note that none of these are acknowledged yet. After, let's say, you send the 100th packet, you receive the acknowledgement for the first packet, then you send another one. You can send as many packets as you want until you fill that pipe. But once the pipe is full, you have to wait for the acknowledgements to arrive. Whenever you receive an acknowledgement, you can remove that packet from your buffer because you know it has been successfully transmitted, and insert a new one into the pipe so that you can fill the pipe once again. So if your data generation rate is faster, instantaneously of course, than your transmission rate, you have packets in hand to be transmitted. Okay? When the pipe becomes a little bit empty, you can inject more packets into the pipe so that you have higher efficiency of the pipe. Of course, in the long run, your data arrival rate should not be higher than your transmission rate, because otherwise you will need infinite buffers on this end. Okay? So in such a system, if you understand that one of the packets in the middle has been damaged or lost or whatever, it didn't arrive in time, now you have to fix this. In the case of go back N, what you do is you go back in time you look at the history of the packets in your buffer, find the last one that was successfully transmitted, and starting from there, start retransmitting. Which will mean that actually you're retransmitting many frames, unnecessarily in fact, because probably they would be properly received, but you go back and repeat all of them. That's called the go back N, as the name implies. Whereas in the case of selective ARQ, you just retransmit the one that's missing and jump back and continue from where you were left. Okay? But these protocols are better in terms of performance, but they require more complex circuitry or more complex mechanisms to implement. ARQ mechanisms are a form of reliability. They provide reliability in contrast to FEC mechanisms, forward error correction mechanisms, in which you don't do retransmission, but in the original message you have sent, you have enough redundancy so that some limited amount of error can be tolerated at the receiver end. The receiver detects that there has been an error, but without asking for any retransmissions, the receiver is also able to fix the error and understand what was actually meant. And this actually happens in real life, in your DNA. In the case of DNA, actually, you have some redundancy available. The efficiency and the effectiveness of such communication protocols typically depends on being tuned to handle the bandwidth delay capacity, as we discussed. For example, in terms of waiting for acknowledgements and handling congestion. Remember, TCP was mostly based on the assumption of congestion. For the case of molecular motors, the channel is essentially a mechanical instrument, because a motor is actually physically walking over that rail to the destination. And the capacity is typically related to the density of the molecular motors. So now going back to that water pipe example, if you can put the molecular motors one after another on the channel, you will increase your efficiency. That's obvious. But remember, we said that when there is a congestion of these molecular motors, when there's a high density of the molecular motors in a given part of the rail, the probability that these will be detached from the rail is higher. Okay? So while we are trying to increase the efficiency, we will end up with actually higher error, which will harm efficiency. So you have to find a good balance of that. So that should be taken into account. But do we really need to always miniaturize what we have in the human scale networks into the nanoscale networks? This should be something you should keep in mind. Okay? So as I said, try to think outside the box. How can we, do we have to retransmit in case of errors all the time? Or may I just let it be errored? Is that an option? Consider that. And also, can we encode the information in a redundant way, like FEC actually, to tolerate errors? Especially if I can ensure that errors are bounded. So, we have already discussed some human-engineered ARQ protocols. Does the nature really do such error control? We should look at that. And does the nature really try to do error control? That's also a question. However, error control is a little bit important, especially if we're talking about the carrying of information using DNA. Because if it's damaged, then you get something different, something unexpected, let me say. So we should make sure that the messages are typically conveyed without error in living organisms in some types of communications, of course. The genetic code is likely to be one of the first biological nanoscale communication mechanisms that comes to one's mind when we discuss about safe transmission of the information, especially between the generations. So DNA plays a significant role in nanoscale communications. One thing is its ability for self-assembly. It can reproduce the packets, let me say, and its ability to act also as a molecular computer. It's not only the messaging mechanism, but it also does processing. And finally, it's a molecular scale transmission medium. So, if you look at DNA in more detail, the information in the DNA is typically encoded as a linear string of letters represented by some chemical bases. There are 4 types of different bases, chemical bases. These are adenine, cytosine, guanine, and thymine. In short, people use A, C, G, and T, the capitals. Adenine and guanine are larger bases. They're larger molecules. Typically, that class of bases are called purines, while the cytosine and thymine are pyrimidines, and they're smaller bases. These letters are used, as I said, for these bases, and these are placed on a sugar backbone which maintains their order, which means they all get attached on a sugar backbone so that they appear as a string of letters. One good thing about DNA is that these bases typically fit together in a good manner. They form a kind of a weak bond, which is actually hybridization. This is a hydrogen bond, and this is a weak but stable bond, and when necessary, by heating it, for example, you can detach it easily. And one good thing is, if you consider these 4 bases, A and T fit well together. Similar for C and G, but for example, A and C or A and G will not fit together. Okay? And these are typically known as base pairs. A and T is one base pair, C and G is another base pair. And these are, as I said, kept together with those hydrogen bonds. That's what hybridization is doing. As you all know, a strand of DNA typically attaches with another complementary strand using mechanisms provided by DNA, and they're able to replicate. And a single strand can serve as a negative template for the other strand, for its complementary strand. And when a new strand is created, according to the complementary strand, a duplicate of the original strand is produced. We'll have 2 video demonstrations. I've prepared the videos here, so let's go over these videos very fast. So this one's composed of several steps. There are also some exercises in between. You have the link, so you do the exercises yourself. So
DNA replication is the process whereby an entire double-stranded DNA is copied to produce a second identical DNA double helix. The objectives of this exercise are to understand the functions of the proteins responsible for DNA replication, including helicase, SSB protein, primase, the sliding clamp, DNA polymerase, RNase H, and DNA ligase, and to understand why the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously. DNA replication is an intricate process requiring the concerted action of many different proteins. The replication proteins are clustered together in particular locations in the cell and may therefore be regarded as a small replication factory that manufactures DNA copies. The DNA to be copied is fed through the factory, much as a reel of film is fed through a movie projector. The incoming DNA double helix is split into 2 single strands, and each original single strand becomes half of a new DNA double helix. Because each resulting DNA double helix retains 1 strand of the original DNA, DNA replication is said to be semi-conservative.
So I'll skip these.
DNA replication requires a variety of proteins. Each protein performs a specific function in the production of the new DNA strands. Helicase, made of 6 proteins arranged in a ring shape, unwinds the DNA double helix into 2 individual strands. Single-strand binding proteins, or SSBs, are tetramers that coat the single-stranded DNA. This prevents the DNA strands from reannealing to form double-stranded DNA. Let's look at the steps of DNA replication in more detail. To begin the process of DNA replication, the 2 double helix strands are unwound and separated from each other by the helicase enzyme. Now that they are separated, the 2 single DNA strands can act as templates for the production of 2 new complementary DNA strands. Before the lagging strand DNA exits the replication factory, its RNA primers must be removed and the Okazaki fragments must be joined together to create a continuous DNA strand. The first step is the removal of the RNA primer. RNase H, which recognizes RNA-DNA hybrid helices, degrades the RNA by hydrolyzing its phosphodiester bonds. Next, the sequence gap created by RNase H is then filled in by DNA polymerase, which extends the 3' end of the neighboring Okazaki fragment. Finally, the Okazaki fragments are joined together by DNA ligase, which hooks together the 3' end of one fragment to the 5' phosphate group of the neighboring fragment in an ATP or NAD dependent reaction. We are now ready to review the steps of DNA replication. The process begins when the helicase enzyme unwinds the double helix to expose 2 single DNA strands and create 2 replication forks. DNA replication takes place simultaneously at each fork. The mechanism of replication is identical at each fork. Remember that the proteins involved in replication are clustered together and anchored in the cell. Thus, the replication proteins do not travel down the length of the DNA. Instead, the DNA helix is fed through a stationary replication factory, like film is fed through a projector. Single-strand binding proteins, or SSBs, coat the single DNA strands to prevent them from snapping back together. SSBs are easily displaced by DNA polymerase. The primase enzyme uses the original DNA sequence as a template to synthesize a short RNA primer. Primers are necessary because DNA polymerase can only extend a nucleotide chain, not start one. DNA polymerase begins to synthesize a new DNA strand by extending an RNA primer in the 5' to 3' direction. Each parental DNA strand is copied by 1 DNA polymerase. Remember, both template strands move through the replication factory in the same direction, and DNA polymerase can only synthesize DNA from the 5' end to the 3' end. Due to these 2 factors, one of the DNA strands must be made discontinuously in short pieces which are later joined together. As replication proceeds, RNase H recognizes RNA primers bound to DNA templates and removes the primers by hydrolyzing the RNA. DNA polymerase can then fill in the gap left by RNase H. The DNA replication process is completed when the ligase enzyme joins the short DNA pieces together into one continuous strand.
Now that would be the end of the first video. Let me go over the second one. This is actually explaining the same thing from a different perspective. This is much shorter. So look at that.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
So this was about the formation and duplication of DNA. Of course, that's not something you're typically responsible for, but you should have an idea about how these things are working to understand the other stuff. So that was some additional information for us. So let's continue with the networking perspective on the use of these. So according to the descriptions we had about DNA replication, it's assumed that there are no errors in this process. A true understanding of the DNA communication channel will require that we understand the mechanisms of coding information on DNA and also the channel capacity. And that further requires that we should understand the DNA communication, what the DNA communication channel is comprised of, and make sure that we have some redundant error coding information so that yes, there will be actually errors, so we should be able to cope with the errors. And one such example is given in 23 with an even parity mechanism. When we have the error coding, our primary goal is to be able to say, able to do this easily and have a mechanism for detecting the errors, the mistakes in the DNA sequence, and at the same time make sure that if an error occurs in part of the DNA, the information written there, when modified with the error, does not resemble some alternative form of information. In other words, when you're encoding the data on DNA, if you look at the possible valid codes, there are some valid combinations and there are other combinations that do not correspond to any kind of information you would encode there. So they would be invalid. So if an error hits, the transformed form of that piece of information should not correspond to an alternative representation, because in that case you cannot detect that there was an error. The error hits, it should make your information have an invalid coding so that the receiver, when it sees that invalid coding, at least it understands that there's an error. So that means in the coding space, two valid codes are well separated so that with few errors, one or two errors, you cannot switch from one of the valid codes to the other. Few errors should take you to only invalid representations so that you can detect the error. So distance between the possible combinations should be far apart. So the lock and key nature of these base pairing in DNA representation, enabled by the hydrogen bonds, is one of the keys that provides this. each of the DNA bases has a sequence of 3 connecting structures. So they're in triplets. Either there's a hydrogen donor or an acceptor to form the hydrogen bond in between. So each nucleotide base can be considered as a sequence of 3 binary values because of these triplets. And the fact that the base is either a pyrimidine or a purine, remember 2 of those 4 keys would be coupled for the pairs. One would be a pyrimidine, the other a purine. They appear to serve as the final pair to bit. Okay, like you don't have A and G together. Okay, that's it. That serves also as a final pair to bit. And during this mapping, the DNA bases can conceptually be placed on non-adjacent corners of a cube. Okay, like consider a cube, you have one here, you don't have the other one at the other end of the link. It has to be at the other corner. Okay, if you place them in such a manner, then a single bit error will not take you to a valid code word, because a single error would be taking you to the other end of the edge, but you don't have a valid code there. You need another error. You need 2 errors together to go from one valid code to the other, for example. There are several types of cellular scale motors other than the molecular motors that ride on the rails. So this is a new, by the way, I just started this slide, but this is a new subsection here. So besides these molecular motors, there are other ways of having molecular motors, not running on rails. The ones that run on rails, like the microtubules or the actins, they appear to be useful for short and very directed transport, because you put the molecular motor on the rail, it goes to the other end of the rail. So it's really directed. Or in the case of actin, the other end of the actin filament is at another location. But there are also catalytic motors that appear to be relatively medium range compared to the molecular motors. And also there are the flagellar motors that are the longest range of communication mechanism using the molecular motors. As always, there's a trade-off. Shorter range rail riding molecular motors are highly directed and targeted, whereas the others, like for example the flagellar driven, are able to go longer distances, but they're not well directed, as we will see the video later. They just go in random and hopefully they will reach at the destination at some time. And as an example of the flagellar motor, we can talk about the Serratia marcescens bacteria. It's a single cell organism and has an elliptical body that is 0.5 micrometer in diameter and 2 micrometer in length. And it has what we call a flagella behind. That is like a tail, actually multiple of those. They are flexible helical structures that are 20 nanometers in diameter. So they're very thin. However, they're 10 micrometers in length. It's like 5 times the length of the bacteria itself. Okay? And bacteria has 4 to 10 of such flagella, like 4 to 10 tails behind, long tails, that helps it to move around. And the propulsion force is approximately 0.5 piconewtons. Recall from our previous discussion on the Reynolds number and the Brownian motion that these little organisms are greatly affected by collision in the environment with the other molecules in the fluid environment rather than having their own inertia. And also, as we will see, the flagella behind will turn in one direction and then it has to rewind. So it will change the direction there. We'll see examples of that. It appears that the force of the flagella propels the bacteria in a random direction. That's important, in a random direction at approximately 47 micrometers for a second, and then it will change the direction and again make a similar movement. Okay? However, though we say it makes a random movement, there are also ways of directing these bacteria. We'll also talk about those. The flagella can switch the direction of rotation, as we mentioned. In the counterclockwise rotation, the flagella, that tail, bundles up into a coil, and this takes the bacteria, in quotes, run in a given direction for some time. But then the bacteria will switch its flagella's rotation in the reverse direction, in the clockwise rotation, and the bundle is uncoiled and the bacteria goes into what is called the tumble. And the tumble lasts for only a fraction of a second, and then it will run again. So the movement of the bacteria is like, it will run for about a second, tumble here, so it will run this way, tumble while it's recoiling, and then the direction has changed. It will run again, tumble, go, tumble, go, tumble, go. It's the movement of this bacteria is actually a sequence of runs and short tumbles in between. Okay, so it is.
These tumbles seem random.
Once again.
These tumbles seems random.
Mostly yes. Mostly yes, but there is a way of slightly directing the bacteria. For example, you can provide some chemicals around the bacteria so that it will try to follow that, but it's limited. So this direction of swimming in the fluid, or running, however you call it, is changed in response to some chemical substances, which are called the attractants, if it's trying to follow it, or maybe it's trying to run away from it. If it's a hazardous chemical, for example, for a bacteria, so it would be, in that case, a repellent, detected in the environment by the cell's chemotaxis system. So if you provide food to the bacteria, it will try to go towards the food, actually. Just as with the rail walking molecular motors we have discussed, cargo can be attached to these organisms with molecular motors, or you can also inject DNA, for example. The paths of the chemical substances which are used for attracting these bacteria can be built to route the cells for data communication. So if you can provide the food beforehand, then you can expect it to follow that food and go in that direction. Or you can also put some obstacles, as you will see in one of the videos. The flagellum is composed of over 20 different proteins, plus 30 proteins in addition for regulation and the assembly of the flagella. And the assembly of the bacterial flagellum can be examined in three main parts, which are the basal body, hook, and the filament, as in here. Okay. So consider it this way. This is the cell membrane. So this is the below you have the internal part of the cell, and this is the outside. Okay. This is the filament and the tip of the filament. So this is like a tail. Okay. And here you have the motor that is rotating that filament. So the filament is just rotating like this, or the other way, clockwise or counterclockwise. So this is the motor part, and this is serving as the hook that connects the filament to the motor. Okay? So the basal body is a rotary motor which consists of a rotor, a drive shaft, a series of bearings, stator complexes, and an export apparatus. So you have the MS ring, which is this yellow ring here. You have the C ring, which is this blue ring here, and the type 3 segregation system here. So inside this, inside these rings, this green thing will rotate actually. The drive shaft is the rod here, which is attached to the filament through this junction. So it's carrying this movement to the filament. And there is a series of bearings like L and P rings, and also mod A and mod B stator complexes, which help the rod to turn inside these rings relative to those rings. Now, we'll again have some short videos explaining how these things are working, then we will be discussing more about this stuff. So, the first two videos are showing examples of this Martensens bacteria. It's a very short one, so I will have to play it over and over. Here you see, okay, I guess there's no way of replaying it continuously. Now these are your bacteria moving. As you see, let me once again show it. It looks funny, but as you see, these things are once in a while tumbling. See how it's tumbling? Okay? I guess that would be enough to replace. And this is somewhat different. Here you have some obstacles in the environment, and you have your bacteria that are trying to move it. Now, there would be some more information provided here about the width of that stuff. So, these channels, they don't fit together on the screen. These channels here, for example, if you look at this one, the width of the channel here is ranging from 5 to 10 micrometers. Some parts of it are narrow, some parts are wider. Okay? Whereas this part is much wider compared to this part. When you're watching the video, this one is slightly over a minute. Follow how fast these bacteria are moving towards the destination in this channel and also in this channel. And look at the pattern of the movement. That's important. Now see, the bacteria here are stuck with these obstacles. So yes, they can pass through, but they're having difficulties in passing through. Whereas in this channel, see, like this one got stuck here actually, but you can see that many of them are able to pass from one side to the other. Okay? So there is this tumble effect, but if the channel in which it's trying to move is wide enough, it can have almost a directed movement. Whereas if it's below some limit, then it's very unlikely that they will reach a destination. Some do, but it's not moving in a more directed manner in that case. Okay? Similar also in here. These are very narrow lines, but these are very well directed.
Do they always move, not stopping?
Well, if it has energy, it does. If you wait there, you will not find any food. You consume all the food around. Either the food should come to you, someone should serve you the food, or you should walk around to find the food. Probably that's not the case for the human beings. Let me stop this. And This shows how the flagella propel themselves through their environment.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rotobacter cells have one flagella each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell surface, make up the three parts of the flagella. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mode A and mode B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mode B functions in proton conductance. Each stator contains two mode B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the mode B proteins. This binding causes a conformational change in mode A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called FLEF come together in the plasma membrane to form the MS ring. The FLE-G proteins assemble under the MS ring. FLE-G, along with FLE-M and FLE-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagella are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mode A and mode B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but mode B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
The hook always in a curved structure.
Once again,
The hook part, it should be curved to make the more.
yes, yes. I mean, even if it's not properly hooked, probably it will, due to the helical structure of the filament, still it should be able to rotate, but having it curved will make it better. So, going over that information, the hook serves as a universal joint between the filament and the basal body, as you've seen. The filament can be viewed now as a propeller connected to the motor with its helical shape. So this just works like the motor at the back of a boat. It is a capping protein at the tip, and the new subunits are added to the tip of the filament until that cap is thrown away. And hook and the filament work together to convert the rotary motion into thrust. And that rotary motion is due to that proton transfer. So the motion is fueled by the inward directed electrochemical flow of the protons, called the proton motive force. So as mentioned in the video, the exact operation inside the flagellar motor is not exactly known, but there are some widely accepted theories. So the protons, as mentioned, flow through the interface between the two types of proteins, which are mode A and mode B, that make up the stator. The stator actually connects the basal body, fixes the basal body to the cell membrane, because if it's not fixed, that part would turn inside the cell without making the flagella turn. Okay, so you have to somehow fix the motor to somewhere. Like if your motor in your car is not fixed, rather than turning your wheels, the motor itself will turn inside your car, which will not move the car. So each stator contains two mode A and mode B proteins, and also two aspartic acids that actually work with this proton motive force. And the protons move through this channel inside the stator. They bind to the aspartic acids in the mode B protein, and then they're released again. So this binding causes a change in the mode A proteins. So it made that movement, which was transferred to the basal body there. So it creates the first power stroke. And at the end of the first power stroke, the two proteins will now be released from that mode B protein, and they will just go into the cytoplasm, and this proton loss causes a second stroke, and it will turn this ring here. So, in addition to the flagellar motors, it's also we can also look at how we can use DNA for such purposes. Using DNA, you can do both computation. You can also construct synthetic molecular motors directly from DNA by creating the proper three-dimensional connections between the DNA strands. The molecular switch component of a motor can also be constructed based on some external conditions like the temperature and ionic concentrations. So we're now able to control the DNA, and this makes an ideal construction material for biomechanical devices. It allows us to develop this technology. The specificity, which is the precise control over the location where DNA will join together, is very high. So by generating the correct nucleotide sequences, it's possible to confidently control what components will connect together or which components will hybridize together. This is already done by the biology world. The DNA has a persistence length of 50 nanometers. So for devices built from short sequence of several nanometers, they can be used. Single-stranded DNA has much lower persistence length, and it's very flexible, so it can serve as the joints. And the restriction enzymes can also be used to cut DNA at specific points. And also the spiral shape of the double strand can be flipped from a right-handed helix to a left-handed helix. You can change it rotation. With a longer and better match can replace an existing strand, causing a conformation change, and that conformation change turns into a mechanical movement when necessary. This strand exchange technique has been used to implement a wide variety of different mechanical devices, like the nanoscale tweezers to hold things at the nanoscale, of course. And the autonomous DNA devices have been created that traverse along double-stranded RNA and can carry a payload, just like the molecular motors do. One other thing is the use of catalytic motors. So a catalyst is a substance that is either increasing or decreasing the rate of a chemical reaction. Okay? So it either eases the reaction or it prevents the reaction. So by making use of it, it's also possible to provide motion, for example, to the cells. So the catalyst generally reacts with the input chemicals to form some intermediate substances that are then converted to the final reaction product. This will help increase, for example, the forward direction rate of the chemical reaction. And one of the most well-known examples of catalysis is the increase in the rate of decomposition of hydrogen peroxide into water and oxygen. If the catalyst is distributed non-uniformly around a cell, for example, then there is an unbalance around the cell. So due to this unbalance, you can create a net force or torque so that the cell turns that way. So if you have catalytic motors around the cell, if you provide the catalyst at one side and not at the other side, the rate of reaction on each side of the cell will be different, so you can make a turn in this manner, and the object will move. So that would be the end of this chapter. So here we go with chapter 3 on gap junctions and calcium signaling, a different type of communication, this time between the cells. What we did in the previous chapter was inside the cell. So the use of flagellar motors enables us to carry the information at longer distances between the cells. The mechanism it uses is the molecular motor. That's why it was covered in chapter 2. But here we're focusing on a method of communication among the cells. In this chapter, we'll be talking about, in general, about what gap junction and cell signaling mean, and discuss where and how calcium signaling is used in nature and how it works. Then we'll focus on gap junctions and talk about the liposomes. Then we'll look at cell signaling and talk about network coding in that respect. We'll talk about modeling signal propagation and diffusion. And finally, we'll talk about a different way of communication between the cells, this time communication via diffusion. And that will conclude the chapter. And probably it will take us about 2 weeks to finish this chapter. In the previous chapter, as I mentioned, we covered the topic of molecular motors for transporting information inside the cell. The nanonetwork mechanisms in that chapter operate typically within the cells, but in this chapter we'll continue based on those concepts to talk about communication between the cells rather than within the cell. Our goal is to focus upon a few potential approaches in depth and then briefly discuss some broader mechanisms. We will start with the explanation of the gap junction signaling inside at the cell membrane. Then we will cover some biological signaling mechanisms over longer distances between some source and destination cells. Typically, cells use several different mechanisms for communication and also for coding, which are not so different from what we are using in the human scale for our engineered cells. There are many examples, but some of them are specifically, they need to be mentioned, like amplitude shift keying and frequency shift keying, frequency modulation using custom signaling. Redundancy and error coding also appear in chemical cell signaling techniques. We'll be talking about them very briefly. Maybe we should first talk about a comparison between the techniques used in the traditional networks as opposed to what we have in the nanoscale networks. So table 1 briefly discusses that. We'll be focusing on these, but let me very briefly explain beforehand. If you look at the carrier, the type of the signal, the propagation speed, environment through which the signal propagates, the information that's being transmitted, the receiver behavior, and other features. If you look at these concepts, in the traditional networks, for example, as the carrier, we use electromagnetic signals, whereas in the nanoscale systems, typically, not necessarily, but typically, molecules are used for carrying information. So if you look at the type of the signal in the traditional networks, again, either electrical or optical signals are being used. In the case of nanoscale networks, mechanical or chemical signals are used. For the case of propagation, since we are using electromagnetic waves, in the traditional networks, we are working at speeds close to the speed of light. Whereas in the case of nanoscale, we're talking about speeds that are relatively much slower compared to electromagnetic signals. The environment in the case of traditional networks is either airborne, or for the virus, or fiber or copper guides. Whereas in the case of nanoscale networks, we're talking about the aqueous environment between the cells, for example. For the information that's being transmitted, in the case of traditional networks, the aim is to provide interaction between the humans, or provide interaction between a human being and the machine. So we try to provide voice, text, or video, whereas in the case of nanoscale networks, at least one of the sides, one of the peers in the communication is in nanoscale. Therefore, we're talking about physical and chemical states of the machines, nanomachines. The receiver, in the case of traditional networks, the receiver interprets the encoded information, whereas in the case of nanoscale networks, chemical reactions occur at the receiver. And in the case of traditional networks, high accuracy is desirable, but that requires high energy to provide that accuracy. However, in the case of nanoscale networks, stochastic states appear. We are not exactly sure about what has been transmitted in most cases, but we try to work at very low energies because these devices typically do not have as much energy as your cell phone, for example. Okay, so let's go over these slides very fast because we have discussed some of this stuff by looking at the table, but the physical medium is completely different. As we said, typically in traditional networks we are using electromagnetic signals. In the case of nanoscale networks, you can work with electromagnetic signals. For example, you can work with terahertz communication. You can have signals propagating at, working at terahertz, but in most cases we're talking about transmission of molecules rather than electromagnetic signals. But then that means communication at very low data rates. The signals, as in here, since we're using electromagnetic signals, we have either electrical or optical signals. That's not our case in nanoscale. We said the speed is different. The distances are very different. In the case of nanoscale networking, we're talking about distances of hundreds of nanometers or several tens of micrometers, typically. Whereas in the case of traditional networks, we talk about kilometers or tens or hundreds of kilometers. The traditional networks are primarily wireless or fiber or copper-based. In the case of nanoscale, we're mostly talking about liquid environments, especially if we're talking about communications, about molecular communications. Traditional human scale networks have humans at one end. So the decoding of the information is intended for the human being as the user. Therefore, we're carrying typically voice, video, or text. Whereas in the case of nanoscale networks, the receiver itself should be a nanomachine. So we're not considering such applications. So coding and decoding should involve chemical reactions. That means a completely different design for transceiver and also the receiver. We're talking about very low energy communications at very low data rates in the case of nanoscale networks, and we have to accept that we're communicating in a stochastic manner. We don't have high accuracy. So rather than using a system of motor proteins, as we did in the previous chapter, now let's also look at the case of signaling molecules that are not necessarily moving along some microtubular rail topology. Okay? So you don't have any rails or deployed infrastructure for communications. The basic method is to make use of this aqueous environment between the cells or the nanomachines. So one of the approaches is making use of some gap junctions between neighbor cells. Pay attention to that. They should be neighbor cells. They should be touching each other. In both cases, the information molecule is released into the medium and propagates through Brownian motion, as we discussed earlier, through the gap junction into the adjacent cell. So for the case of intracellular communications, we could be considering two different approaches. The one we will focus at the moment will be the one in part A. By the end of the chapter, we'll be talking about part B. So in this one, we have a sender and a receiver. So this is encoding the information, sorry, and this is supposed to receive that transmission and decode it properly. Okay? And these cells are neighboring cells to each other, and at these points, you have some gap junctions previously formed between these cells. The cells are able to construct gap junctions in between, and once they're constructed, the cells are also able to open and close them as they wish. And this can be done in a very short duration. And each gap junction has a given lifetime. So what we are trying to do with the, in the case of calcium signaling is this encoder creates a waveform that has some information encoded on it using some feature of the signal, like the frequency, amplitude, whatever. You should be able to encode your information, let's say ones and zeros, on this feature of the signal, and the signal should somehow propagate to the destination. Okay? So that's what we're trying to provide. But we'll see that actually we're not passing the calcium itself. Although the name is calcium signaling, this looks like you're sending the calcium from here to the destination. That's not the case. Actually, each cell keeps its calcium. So the calcium ions here will not be moving here. Very few of them will just leak through the gap, but they can be ignored. The important thing is the concentration of the calcium inside the cell will be fluctuating in a controlled manner, so that the signal is propagated, but actually the calcium ions will not be moving around, but some other molecules will be moving around, as you will see. As opposed to an alternative approach, which, as I said, we will be discussing by the end of the chapter, is through diffusion. In that case, you release the molecules from here, and they will propagate to the destination with Brownian motion, and they will be received there. Chemical signaling among these cells is the basis of this approach. This is largely biological in nature, and this approach can take advantage of previous work from biology to understand the best way to approach this challenge. The cells can use three methods for signaling. One is they can secrete chemicals that signal another cell. This way you're triggering some other cell about the information you're trying to send. Or they can contain some plasma membrane-bound signaling molecules that influence the neighbor cells that make direct physical contact. So they should be really in physical neighborhood. And they can form gap junctions that directly join the cytoplasms of the communicating cells. So two cells that are side by side, they form gap junctions in between, and when they open up the gap junction, actually they're opening holes in the cell membranes. That way their cytoplasms will be connected. However, that connection is very selective. It doesn't let everything pass through. We'll see those. So the cells may form gaps in between that will allow the passage of small molecules from one cell to the other. And these communicating gap junctions can be divided into two types. One is the gap junctions that allow molecular passage, and the other is the chemical synapses that communicate via indirect communication between two neurons. Of these two mechanisms, the gap junction has been the method most explored in the literature, and it has been studied for molecular-based communication networks. The gap junctions are typically found in most animal tissue cells, including us, and allow small water-soluble molecules to pass directly from one cell to another. The important thing here is they allow the passage of small molecules, not large ones. The estimated sizes of these gaps are about 1.5 nanometers, and this puts a limit on those molecule sizes that can pass. These small molecules, like the amino acids or the nucleotides, they may be used for triggering the neighbor cells, but larger nucleic acids and polysaccharides typically would not be able to pass through these narrow gap junctions. It has also been shown that the permeability of the gap junctions can be controlled by the cell by making use of the pH or calcium ions and also the polarity on the cell membranes. So this is a useful ability for the cells to communicate with their neighbors. Currently, the most actively research area is on this topic, and it allows local chemical mediators that most cells in the body can secrete to communicate. And there are two methods. One is hormone secretion by cells within the glands, and the other is the use of neurotransmitters. Multiple methods can also be used to simulate a cell to generate these signals. Two of the most common methods are the control of the intracellular concentration of the cyclic adenosine monophosphate, AMP, or CAMP for cyclic one, and the control of the membrane-bound gated calcium channels. We'll be discussing those soon. And the CAMP is synthesized from ATP near the cell membrane. The cells use some special receptors, especially in the case of CAMP, which are proteins that bind to specific signaling molecules, giving the cells the ability to respond to signaling molecules. So what happens is the following. On the cell membrane, there's a specific receptor which is sensitive to messages coming from outside. When a messenger molecule is received by that receptor, the protein here will be activated, and that will trigger a process inside the cell, which will affect the calcium concentration. Then, this is how this calcium wave is initially triggered. Then, that cell will cause a similar reaction, a similar process in the neighbor cells by using one of the two methods, either by sending a specific molecule through the gap junction, or again sending to the external medium a similar triggering molecule which will do the similar process in the other. We will see how that works in more detail soon. This high affinity for signaling molecules is important in molecular-based neural networks, and it allows specific molecules and concentration of the molecules to be used for encoding the information on the signal. Another important feature is the ability for the cells of the same type to be engineered to give differing responses to a specific signal. There are different ways of doing molecular signaling, including the use of signaling molecules that can cross the plasma membrane of the cells, that's the outer membrane of the cell, that can only bind to the surface or a combination of the two, as we discussed, and the signaling molecules move from one cell to another via a diffusive process through the extracellular media, as I mentioned. It's a second method. Or in the case of the gap junctions, diffuse through the connecting cells, as the first method I just explained. And this process can be modeled using Brownian motion. The basic communication method using cell signaling takes place as follows. The information is encoded using an information molecule. So the reception of that information molecule should mean something to the cell. And the calcium is currently one of the most widely used information molecules, whereas the others, like potassium ions, could also be used. Transmission begins when the nanomachine is externally stimulated. One of the nanomachines is externally stimulated, and the information molecule is then propagated from one cell to the other, so that it creates a wave transmission from one cell, from the source towards the destination, and that will be typically done by sending those messenger molecules using Brownian motion either through the gaps or through the extracellular environment. And once those are received at the other cells, they will be repeating the same stuff. Much long ago, cells have gone through the evolution for working in coordination. This has actually allowed for the multicellular organisms, because in the case of a multicellular organism, each cell should not be working independently, but they should be working in a harmonized manner. So there was a need for coordination. And the messenger molecules were secreted to form a channel to carry the information in between. Calcium, phosphate, potassium ions have become the ubiquitous channels for sending these messages. The binding of these messenger molecules, the calcium or the phosphate, changes both the shape and the charge of the proteins, and that is the triggering mechanism in these cells. More specifically, the phosphorylation is the binding of a phosphate group, PO4, to a protein or some other organic molecule. And the cell signaling is also a signaling transduction. So many enzymes and the receptors are switched on and off by the phosphorylation and dephosphorylation, by the binding of the phosphate group or leave of the phosphate group. But if we look at the case of the calcium ion, the cells use a considerable amount of energy to control the calcium concentration inside the cell. And the cell must constantly pump it outside the cell if the concentration inside the cell has gone beyond some threshold. And many proteins have evolved within the cell that bind with the calcium ion in order to buffer it. Or you can also trigger these proteins to release those calcium ions. So actually, there are two well-known calcium pools inside the cell, the endoplasmic reticulum and the mitochondria, that are working as calcium pools that are able to buffer calcium. So you can trigger these organelles to release the calcium ions they have buffered. This way the calcium ion concentration within the cell will increase. Then you can again trigger them to buffer them once again. This way you will decrease it. So what happens is the calcium ion concentration is low, then you increase it, then you decrease it once again. You can increase and decrease in a controlled manner. Now, if your cells side by side, if the calcium ion concentration increases in my cell and then decreases, meanwhile I'll trigger my neighbor. So that in that cell, again, the calcium ion concentration increases and decreases. This way, this is like having the Mexican wave in the stadium. See, you've seen this many times in soccer games, for example. People, the spectators are just sitting in a very controlled manner. Someone gets up, while he's sitting down, the guy next to him gets up, and while he's sitting down, the next one, the next one, the next one. This way, you see a wave going through the stadium, right? The same thing can be done where the person standing up or sitting down is like the calcium ion concentration. So you're able to propagate information from one end of the stadium to the other end, like your happiness, whatever. So the ATPase pumps continuously push the calcium ions into the endoplasmic reticulum or out of the cell. These are called calcium pumps. ATPase is a class of enzymes that catalyze the decomposition of ATP into ADP, which releases a phosphate ion, phosphorylation. So inside the cell, they're voltage gated, that means controlled by the voltage. Voltage gated calcium selective channels, that means channels that only allow the passage of calcium. So there are channels that allow only the passage of calcium ions, and you can control the opening and closing of these channels by voltage. So these voltage gated calcium selective channels that are able to conduct approximately 1 million calcium ions per second, yielding a greater than 10-fold change in the intracellular calcium levels within milliseconds. That's pretty fast compared to some other techniques we will be discussing. These channels appear to be highly selective, allowing only calcium ions to exit the cell through the cell gates, Of course, once in a while, other small molecules may also pass through, but they're negligible. And the selectivity appears to be controlled by the presence of external calcium ions. So depending on the concentration inside and outside, you can arrange that. When the external calcium is removed, that means the concentration outside is decreased, the cell gate becomes less selective and allows potassium or sodium to exit as well. These gap junctions are real physical channels. They're physically there between the adjacent cells, and the cytoplasm within each cell is directly connected when these gap junctions are opened. Having a gap junction is not sufficient, by the way. It has to be open, because it has the open and the closed states, as you will see. So the channel itself is comprised of a pore, of a hole, that is surrounded by hexamers of a protein called connexins. And the connexin forms around the pores of both adjacent cells. In other words, both cells have their gaps made out of connexins, and they're aligned. The two pores should come side by side so that you have a connection between the two cells. And if they're both open, a direct flow could occur from one cell to the other. So it's like this. There are multiple representations of this, but consider this layer above to be the cell membrane of the cell here, and this is the cell membrane of the cell here. Just a small portion of it is in focus. So this one has its gap junctions here, and this one has its gap junctions aligned so that the cytoplasm here can be connected to that cytoplasm as long as one or some of these gap junctions are open. Each one of these is actually a structure like this. Like this is the lower layer here, the membrane of the lower cell, and this is the membrane of the upper cell. And these are the connexin molecules. Together, they form the connexons for cell A, the first cell, or the top cell, and similar for cell B. These, for example, here you see it in the closed form, but they can be opened like the objective of a camera, and this is an open connexon or gap junction. Okay? There are more than 20 different types of connexin proteins found so far, and each imparts different physical properties upon the intercellular channel, depending on with different permeability and selectivity. The channels are dynamic in terms of controlling their permeability and selectivity, so you can open and close them as you wish, and they can respond to many internal and external factors. A primary purpose of the gap junctions appears to be the propagation of calcium as a signaling mechanism, but calcium is not the only one, as we mentioned, but it's the most well-known one. And the calcium waves are induced and appear to be signal information relevant to cell processes, such as the cell growth and also the death of the cell. Okay? The cell is able to create a gap junction on any side it wishes. The cell is somehow able to understand its north, south, or up, down, however you call it, left, right. It can differentiate between these. So it can form its gap junction with any one of the neighbors it wants. The lifetime of a gap is typically several hours. So once you form a gap, it's there for several hours. But you can open or close it in very short durations, to micro or milliseconds. Okay? So you create it, it's there for several hours, but you can open or close whenever you wish in a very fast manner. This allows actually very high data rates for us. The wave nature of the calcium concentration fluctuations is intriguingly like the carrier wave of amplitude or frequency modulated signals we know in our traditional networks. And in terms of signal transduction, calcium signaling appears to be a widely used second level messaging system. In other words, a signal originating from one signaling technique that's translated into a calcium signal for further propagation, because we'll not be sending the calcium itself. Something else will be doing this for us. And the fact that the calcium signal appears in the form of waves indicates that there's a sophisticated encoding mechanism in nature inside the cells, rather than simply binary on-off signals. So this is a very brief demonstration of how calcium signaling works on the one type. What you can do is, we will have this in more detail later on, an outside coming signal, messenger molecule, will trigger the release of IP3 molecules here, and this IP3 molecule will trigger the calcium pool here, like the endoplasmic reticulum, which will release the calcium it has. So the concentration of calcium inside the cell will increase. And this will trigger the creation of more IP3 molecules. And some of these IP3 molecules, with Brownian motion, they will lead through these gaps, if they're open of course, to the neighbor molecule. Meanwhile, the concentration of IP3 molecule will go beyond the threshold, which will now cause a reverse operation. And now the calcium will be stored back in the calcium pool to be used later on again. And this will reduce also the IP3 concentration here. So the concentration of calcium in the cell will first increase, then decrease. But meanwhile, the passage of the IP3 molecules here will cause a similar operation here, and this will do the similar thing in here. Another way of signaling to the neighbor cell would be sending ATP from here, which will diffuse in the environment with Brownian motion and hopefully arrive at one of the neighbors. This time not necessarily the next one, maybe to the other one, we don't know. But the arrival of ATP there will cause a similar operation here, which will trigger others. Okay? So let me stop here for today, and next time we will continue from this point, and we'll be looking at into more details of calcium signaling.